All right, so we are on the cardiovascular system, chapter 18.3. We're talking about the blood, and now we're going to talk about the formed element, the erythrocytes, very specifically. So the erythrocyte, we have already mentioned. The erythrocyte is the name we give to red blood cells, or red blood cells are really the name we give to erythrocytes, or RBCs, shorthand there. Is the most common formed element by far. We've already looked at the hematocrit earlier. A single drop of blood contains millions of erythrocytes and just thousands of leukocytes. So when we're comparing, remember leukocytes are white blood cells. So red blood cells versus white blood cells. There are millions of red blood cells and just 1,000 white blood cells. Specifically, males have about 5.4 million red blood cells per microliter. That's that little U-ish L of blood. And females have approximately 4.8 million per microliter. Again, those are differences in sizing, menstruation, different kind of body styles, amount of muscle, things like that. So in fact, erythrocytes are estimated to make up about 25% of the total cells in the body. So if you remember in any of your biology classes really talking about the trillions of cells in the body, 25%, a quarter of our cells are probably red blood cells. All right, so they're quite small. If a single drop of blood has millions of them, you can imagine they're quite small. Their mean diameter, their average diameter is about 7 to 8 micrometers. Little u again, micrometers, micrometers. The primary functions of the red blood cells are to pick up inhaled oxygen from the lungs and transport it to the body tissues. And then what they do is they do the reverse in a way. They pick up carbon dioxide from the tissues and other wastes, and then they transport it to the lungs for exhalation. So the main function of the red blood cells, of the erythrocytes, is to pick up the oxygen, deliver it to where it needs to go, pick up the waste, deliver it to where it needs to go, mostly the carbon dioxide, to the lungs for exhalation. Erythrocytes remain within the vascular network. So the reason that's mentioned is because the red blood cells generally stay in there. Of course, some of the differences are when there are some issues, but mostly they stay in there. And then the reason that's mentioned is because leukocytes, or the white blood cells, typically leave the blood vessels and then perform their defensive functions. So the movements of the erythrocytes from the blood vessels is abnormal. So that doesn't typically happen. But the white blood cells, it's expected that that will happen. They're supposed to kind of go around and root around and find the bad guys and get them, versus the red blood cells, which are supposed to stay in the vessels and really get the oxygen to where it needs to go and get the waste where it needs to go, which is out of the body. So here's another nice chart. We're going to go over these different pieces. Right now we're at this section, right? The erythrocytes are the red blood cells, RBCs. We talked about 5.2 million in a microliter about. Now we're going to talk about kind of these sections and continue on from there to talk about all of the rest of this. So we can move past this for now. We've talked about the different things that we have already talked about, and then we're going to talk about the shape and the structure of these erythrocytes. So here is a picture of one, and it shows them. They are biconcave discs. I guess we could start up here. So the shape and structure of erythrocytes as it matures in the red bone marrow. Remember, that's where we have hemopoiesis, which is making the red blood cells, making the blood. As an erythrocyte matures, so it extrudes its nucleus and most, right here, of its other organelles. So unlike many of the cells in our body, remember when we are in some general bio classes, 271, we talk about all the different parts of the cell, the Golgi apparatus, the ER, all of these things, ribosomes, mitochondria. A red blood cell really doesn't have all of those things. Here it even says it extrudes, so it gets rid of its nucleus and most of the other organelles. So during the first day or two that it's in circulation, the immature erythrocyte is known as the reticulocyte. So that's important for you to know, that a reticulocyte is an immature red blood cell. And a reticulocyte still contains some of the remnants of the organelle, so it hasn't quite purged or expelled or extruded all of those parts yet. So reticulocytes should comprise about 1 to 2% of the red blood cell count. So not of the total blood, but just of the red blood cell portion, 1 to 2% will be the reticulocytes. So that's about how many, the percentage, that is an immature cell that is going through the process of maturing. And then that provides a rough estimate of the rate of red blood cell production. So when there are abnormally low or abnormally high rates of these reticulocytes, they can look at them, they're diagnostic tests, then they can tell the person that's looking at them, the laboratory person, what's going on. So if there are low amounts, we can take a look at something related to the inability for the production of these cells. If there are high amounts, we can look at why there are high amounts. That really kind of tells the process of red blood cell production, because if it was just all mature red blood cells, we might not know which one is an hour old or which one is 2 days old or anything else. So that really is a good measurement. So let's see, where are we? We are up here indicating deviations. These remnants, primarily of networks, which is where the reticulum comes from. Remember the endoplasmic reticulum? It's a network of stuff there. These networks, or the reticulum of ribosomes, are quickly shed. So as a reticulocyte, they shed these things and mature. And then we have our mature erythrocyte. That has very few internal cellular components. So it doesn't have a mitochondria, which means it relies on anaerobic respiration. So ana meaning without, aerobic oxygen, so without oxygen respiration, breathing. So this means that they do not utilize any of the oxygen that they're transporting. And that's really an important aspect, because if a red blood cell itself needed oxygen, perhaps it would then use up all of the oxygen it just gained from the lungs. So if you can imagine, there's this blood cell, and it also uses oxygen in order to continue living. If it goes to the lungs, picks up oxygen, and then it starts transporting itself, it starts flowing through the blood to get to the toe, right? The tip of the toe coming from the heart up in the chest. That's a really long way for this little tiny blood cell. And if along the way it needs oxygen, maybe it wouldn't have enough oxygen once it gets to the toe. So it is anaerobic. It doesn't utilize the oxygen, so they can bring it all to the tissues. They don't have to use any of it. They also lack the endoplasmic reticula, and so they don't synthesize proteins. What they do, however, is contain some structural proteins. So remember some of those structural proteins, the cytoskeleton, to maintain this shape. This biconcave shape. And that's important because this cytoskeleton, this cytoskeletal protein element, as it's called, the main one is spectrin, the protein spectrin, that allows them to change their shape. So when we talk about the blood vessels, what you'll notice are we have these really large blood vessels that are like huge pipes coming directly off of the heart, where there's a lot of flow. And it goes down, it gets smaller and smaller and smaller and smaller, meaning smaller in diameter, until we get to things like the tips of our fingers, or right at the surface of our skin, even on our stomachs or chest or something. Once you get more superficial in the body, they get really, really small. These are the capillaries. And the capillaries are very, very tiny. So the red blood cells, in order to get to the very, very tips of everything, have to bend and fold and kind of squish on themselves to get to these really tiny areas to then get oxygen to those cells. So these structural proteins are important for that aspect. So erythrocytes are biconcave discs. So they are, plump at their periphery. So the outside here, right, you can see how that's plump in this area. And then very thin in the center, this area here. So it's kind of like a donut that didn't get punched all the way through, very thin in the middle. Since they lack most organelles, there's more interior space for the hemoglobin molecules. And we're going to talk about hemoglobin in just a moment down here. And then that allows them to transport gases, having hemoglobin. So this biconcave shape also provides a greater surface area. So if you can remember, and we'll really see this in the digestive system, where if you have greater surface area that's going to allow for greater movement of substances. So it's important that they have greater surface area so that gas exchange can occur at a greater rate. So in the capillaries, the oxygen carried by the erythrocytes can diffuse into the plasma and then through the capillary walls to get to the cells, whereas some of the carbon dioxide produced by the cells as a waste product diffuses into the capillaries to be picked up by the erythrocytes. So that's pretty basic. We get it, the oxygen comes off of it, and then the CO2, carbon dioxide, goes onto it. Capillary beds are narrow. We talked about how it has to fold and squish, but the space within the capillaries is so small, they have to fold on themselves to get through. So fortunately, those proteins, again, like spectrin, are flexible, so they can bend on themselves. So let's go down here and talk about hemoglobin. So that's kind of the generalized, this is a red blood cell. Now we're going to talk about what a red blood cell does, and really the heart of the red blood cell, or the erythrocyte, and that is hemoglobin. So hemoglobin here is a large molecule made up of proteins and iron, and you can see there are a couple different ways to show that. So it consists of 4 folded chains of protein called globin, okay, designated alpha 1 and 2, beta 1 and 2. So alpha 1, alpha 2, beta 1, beta 2. And each of these globin molecules is bound to a red pigment molecule called heme. So that's where the hemoglobin comes from. So you can see that here. So here's, for example, beta chain 1, and that is this part, this kind of squiggly thing here is a drawing of what a protein might look like. And then this little area here, you can see they have it shown down here. It's maybe easier to draw here. You can see this kind of disc-shaped thing is the heme molecule, and then the iron, that's the iron right there, Fe2+, is attached to the heme molecule. So when we have hemoglobin, that is this whole thing. This is a molecule of hemoglobin. This right here is a protein chain, specifically beta chain 1, and for example over here is alpha chain 2, and attached to this protein, this is the long protein part, is our heme group that then has iron attached to it. And right here, see, this is saying that this is the heme group, and then here is our iron right in the middle attached to the heme group. This would be like the disc. And then that is the red pigment. So the heme group here is what kind of gives it its red color that we talked about with the blood cells. So down here we're talking about each ion in the heme combined to 1 oxygen molecule. So we've got this ion here of iron, and then we've got an oxygen molecule that is able to attach to that iron. So here, this is important, therefore each hemoglobin molecule can transport 4 oxygen molecules. So 1, 2, 3, 4 on each iron. An individual erythrocyte may contain about 300 million hemoglobin molecules, and therefore can bind to and transport up to 1.2 billion oxygen molecules. So we're talking about one single red blood cell can contain about 300 million of these guys, which means it can bind and transport up to 1.2 billion oxygen molecules. So that is a lot of work for that little guy. In the lungs, hemoglobin picks up oxygen, right, which binds to the iron ions, forming oxyhemoglobin. Oxy meaning oxygen, and then hemoglobin, so it's hemoglobin that contains oxygen, so it's picked it up from the lungs. The bright red oxygenated hemoglobin travels to the body tissues, right, where it releases some of that oxygen, and then it becomes darker red. So then once it has released a lot of that oxygen, then it's called deoxyhemoglobin. So D meaning without, right, D, that kind of D. D, and then oxy meaning oxygen, and then hemoglobin. So hemoglobin that does not contain oxygen. Sometimes referred to as reduced hemoglobin. It's been reduced, if you remember your chemistry. Oxygen release depends, right here, depends on the need for oxygen in the surrounding tissues. So hemoglobin rarely, if ever, leaves all of its oxygen behind. So It just depends on how much oxygen a tissue needs. So if one were exercising, the muscle tissue would probably need more oxygen versus another type of tissue. If you're just relaxing, then maybe your muscle tissue needs just as much as your liver, for example. So rarely ever leaves all of it behind, though. So a single drop of blood is not going to drop off its 1.2 billion oxygen molecules to a single cell. It's going to kind of trickle off of them, which is important, right? Because if we are, let's say, traveling all the way to the tip of our toe, for example, again, and we have these 1.2 billion oxygen molecules, and if a red blood cell were, or if all the red blood cells, right, were to kind of purge them all of these billions, all at the very beginning, how would we ever get the oxygen, to our toe? Or how would we get it back, when we're taking the veins, which we'll talk about, back up to the heart? What about all of the tissue surrounding the veins? They would not get the oxygen that they need to continue surviving. So it's important that it kind of trickles off of these heme groups and doesn't just dump it all right away, so that we can get it everywhere. So in the capillaries, carbon dioxide then enters the bloodstream, and here about 76% dissolves in the plasma. The carbon dioxide now we're talking about, we're at the level of the tissue or the level of the cell, and we have gotten rid of some of the oxygen. We're at deoxyhemoglobin stage now. That's what it is. The hemoglobin is deoxyhemoglobin. And now we're talking about the carbon dioxide, which is a waste product, remember, of tissues or of cells. So 76% of that is going to dissolve in the blood plasma. Some of it remaining as dissolved carbon dioxide, and then the remainder forming bicarbonate ion. So about 23 to 24% of it is going to bind to the amino acids in the hemoglobin. So remember, the hemoglobin is going to then pick up that carbon dioxide and then bring it back to the lungs so that we can breathe it out. And then when that happens, when that carbon dioxide is attached to the heme groups, then it is carbaminohemoglobin. Sorry, that's a really long one for me. Carbaminohemoglobin. So as you can see, we've got our hemoglobin aspect of that, and then carbamino, meaning for the carbon dioxide, attached to the amino groups. Remember, an amino group is part of a protein, so it's attached to some of these amino groups in the proteins here. From the capillaries, the hemoglobin carries the carbon dioxide back to the lungs and breathes it out. So changes in the level of the red blood cells can have significant effects on the body's ability to deliver the oxygen. Ineffective hematopoiesis, they changed it up there, went with hematopoiesis. Remember, that's the same as hemopoiesis. I'm not sure what just happened with all of my lines there, but at any rate, here we go. We were here. Ineffective hematopoiesis results in insufficient numbers of red blood cells and results in one of several forms of anemia. So we've talked about anemia a couple times. If we don't have appropriate hemopoiesis, the production of the red blood cells, or insufficient numbers of red blood cells, then we have a type of anemia. On the other hand, an overproduction of red blood cells produces polycythemia. We mentioned that earlier as well. So those two are going to keep coming back, and you're going to need to know those, and we'll talk more about them. The primary drawback with polycythemia is not a failure to directly deliver enough oxygen to the tissues, but it's actually that there's an increased viscosity of the blood. And again, we're going to talk about this a little bit more. They kind of briefly mentioned a couple times, but polycythemia, it seems like it's not going to be a big issue to have too many red blood cells, right? Remember, polycythemia is an overproduction. So thinking to yourself, well, wouldn't overproduction be great because then you have lots of oxygen? Isn't that kind of what blood doping is? Which is true. But then what can happen with that in that case is that we have then that increase in viscosity. And if you can imagine, if you have thicker blood, that can cause some serious problems in the body. So thicker blood is going to lead to higher blood pressure. Higher blood pressure is going to be putting pressure. If you can imagine, these are tubes, kind of soft tubes in the body. And if we have really thick blood and we're really pushing on those tubes, it will just burst, and then that will be a serious issue. In patients, and we'll talk more about that again, with insufficient hemoglobin, the tissues may not receive enough oxygen. So that's the issue with anemia, right? If there aren't enough red blood cells, then there isn't enough hemoglobin, then there isn't enough oxygen being transported to the tissues. But even if the body's making the red blood cells, if you don't have the things you need to make hemoglobin specifically, like let's say you are iron deficient, if you don't have that hemoglobin, then they still might not receive sufficient oxygen, because you can't transport oxygen if you don't have the iron you need to attach the oxygen to. And then that's another form of anemia. So in determining oxygenation of the tissues here, the value of greatest interest in healthcare is the percent saturation. So the percentage of hemoglobin sites that are occupied by oxygen, because that's really the point, right? It's not necessarily how much red blood you have, how many red blood cells you have. It's if those red blood cells are doing what they need to be doing. And the only way we can know if they're doing what they need to be doing is if they have the oxygen attached to the heme groups, and those heme groups are in the red blood cells. And that's referred to as percent sat. So percent saturation is normally monitored using the pulse oximeter, which is applied to a thin part of the body. So maybe sometimes you've seen they put that little thing on the patient's finger. You clip it on to the patient's finger, and then it sends some different wavelengths of light through there, and then it can measure these things. So hemoglobin absorbs light differentially depending upon its saturation with oxygen. So the wavelengths that are going through are either going to be absorbed or they're not, and then it's going to measure that absorption or transmission, and then it can tell, the machine will do calculations and come up with the percent saturation or percent sat. So a normal pulse oximeter reading ranges from 95 to 100%, right? So lots of oxygen attached, everything's going well. Lower percentages reflect hypoxemia, otherwise known as low blood oxygen. Hypo is low, right? And emia is like the oxygen, or is blood, right? Blood oxygen, so hypoxemia. The term hypoxia is more generic, so you might have heard hypoxia, and it just kind of refers to low oxygen levels, which is something that can happen generally. So somebody might be in a state of low oxygen levels for whatever reason, maybe just exercising, and then they have hypoxia, but that doesn't mean that they have hypoxemia. That is more of a disease-related route. So oxygen levels are directly monitored from free oxygen in the plasma following an arterial stick. So that's another way to do it. So there's the kind of general easy route of doing the clip on the finger, and then there's an actual arterial stick, meaning a stick in the artery pulling blood, and then they can measure the amount of oxygen. So this part talks about when this method is applied, the amount of oxygen is expressed in terms of partial pressure of oxygen, or simply this PO2, and is typically recorded in units of millimeters of mercury, which is this mmHg. So those are two different ways to measure oxygen, whether or not it's attached to the heme groups that are getting to the tissues. So the kidneys filter about 180 liters of blood in an average adult each day, or about 20% of the total resting volume of blood. So this is a really great spot. So the blood is going through and going through and going through, so about 180 liters a day are going through. And then this is a great spot then for receptors to determine oxygen saturation. So our body, again, wants to be in hemostasis, which is, balance. And so they have receptors, the bodies have receptors in the kidney that are going to be calculating, basically, the oxygen that's coming through, and have receptors for whether or not there is a high saturation of oxygen or a low saturation. So in response to hypoxemia, remember hypoxemia is low blood oxygen, hypo meaning low, less oxygen will exit the vessels supplying the kidney. So then let me kind of scroll this up here so we can read the rest of the paragraph here. So in result, again, to hypoxemia, less oxygen will exit the vessels supplying the kidney, resulting in hypoxia in the tissue fluid of the kidney, where the oxygen concentration is actually monitored. So what that's saying is that if the blood or if those heme groups don't have enough oxygen attached to them, Because of the way osmosis works, or because of the way the flow of things across membranes works, right? It goes from an area of high concentration to an area of low concentration. The blood cells will have low concentration, which means that it won't transfer as much oxygen into the tissue fluid of the kidney. And then that's where it's monitored. It's saying, okay, if we don't get a lot of oxygen from these blood cells, then that must mean there must not be a lot of oxygen on the blood cells to give to this tissue or this fluid. So the interstitial fibroblasts here within the kidney will then remember EPO. What was EPO? Erythropoietin, right? So it secretes EPO or erythropoietin. Remember, what did erythropoietin do? Erythropoietin increased red blood cell production. So we have our kidneys that are saying, okay, we're not getting enough oxygen in this fluid. And then, that was a little test for you, by the way, but you're not getting enough oxygen in the fluid there. And then it says, okay, there must not be enough of red blood cells. There must not be enough oxygen in the red blood cells. So what we need to do is we need to secrete this erythropoietin, get it out there so it can get to the red bone marrow so that it can start producing more red blood cells, so we can increase our oxygen in the blood. So then here, increase, you could have cheated on my little quiz there, increasing erythrocyte production and restoring the oxygen level. So get us back into that balanced mode. And again, remember from the beginning of the chapter, we said that it's on a negative feedback loop. So that's what that's talking about. As oxygen then saturation rises, so our EPO has gone to the red bone marrow, said make more red blood cells. We make more red blood cells. The blood cells are picking up oxygen from the lungs, getting all the way to our kidneys. Our kidneys say, okay, great, that's a good level. So then we have our oxygen saturation has risen. Then our EPO secretion is going to fall. Erythropoietin is no longer needed because our oxygen levels are good, and then vice versa. So that's how it maintains homeostasis. And then it has a little note here about populations at high elevations. So it's mentioning that if you've heard of altitude sickness before, so populations dwelling at high elevations, higher elevations have lower levels of oxygen in the atmosphere, so what they're breathing in has less oxygen. And so they naturally maintain a hematocrit that's higher than people living at sea level. Remember, hematocrit, right, was the amount of red blood cells. So they have more red blood cells, a higher hematocrit than people living at sea level. So if they have more red blood cells, what does that mean? That means that they are going to be able to transport more oxygen. They're going to have more red blood cells going through the body so that they can have more red blood cells picking up oxygen as much as they possibly can. Consequently, people traveling to high elevations may experience symptoms of hypoxemia. So that's where the sickness comes from. Fatigue, headache, shortness of breath, as soon as one arrives to that high level. In response to the hypoxemia, then, the kidneys are going to secrete EPO, just like it would at a normal time, to step up that red blood cell production until we get to homeostasis again. So in order to avoid the symptoms of hypoxemia or altitude sickness, mountain climbers usually rest. You know, they get to one point and then they rest for a while, a couple days to a week, so that they can get that kind of figured out, so that the body can secrete that EPO, so that they can increase their red blood cells, so that they can keep going. But in cases when they're climbing the tallest peaks like Everest and K2, the second highest one in the Himalayas, they may rely upon bottled oxygen because it just takes so much time for the body to produce that many red blood cells. And then at that point, remember, we get to the polycythemia, which is where we have so many red blood cells that our viscosity gets really thick, and then that can take a real big toll on the heart and trying to pump all of that really thick blood through. So then we can talk about our life cycle. I don't know why those are disappearing. That's kind of a bummer, but we can talk about the life cycle of the erythrocytes. So the production of erythrocytes in the marrow occurs at the staggering rate of more than 2 million cells per second. Remember, it's very important that we have our red blood cells. They don't last for all that long. We need to have this rotating amount of cells coming in and dying and then being processed. And so for this to occur, a number of raw materials must be present in adequate amounts. So we have to have all of the things that we need, right, to piece all that stuff together to make those 2 million cells per second. These include the same nutrients that are essential to the production and maintenance of any cell. So things like glucose, lipids, and amino acids, but there are some that are specific to the red blood cells. And these are the trace elements. So I suppose you could probably guess the first one, which is iron, right? We have to have enough iron to be able to make these heme groups with the iron on them to attach to these heme groups. So we've said that each heme group in a hemoglobin molecule contains an ion of the trace mineral iron. On average, less than 20% of the iron we consume is absorbed. So it's very important to make sure that you get enough iron in your body so that your body can keep making these red blood cells. And so it's important, again, that we have all of these pieces that we can stick together to make the end product, which would be the red blood cell. Here it mentions, just really quick, heme iron from animal foods, meat, poultry, fish, is absorbed more efficiently than non-heme iron from plant foods. So it's actually easier for our body to absorb these things, but that is actually kind of contested, whether or not it actually is easier and whether or not it is actually safe at those levels. And it's kind of a situation of when so much is coming in at one time, the body isn't able to process all that. So it may be easier to get it in, but then you're not actually getting as much of it because the body is kind of overloaded with the heme iron from animal foods versus the plant foods with the non-heme iron. It comes in a little bit more slowly. The body has to process it, so it's not absorbed as efficiently, but it can be shown that the trickle-in method is actually sometimes better, rather than completely flushing everything with this heme iron to where the body can just grab it what it can grab. The plant foods, while it's processed and absorbed slowly, it perhaps is able to grab more of it because it can grab a little here and then a little bit here and a little bit here. So that's kind of is being looked at a lot. So anyway, that's the side of nutrition as a side note. But upon absorption, the iron becomes part of the body's total iron pool. The bone marrow, liver, and spleen can then store the iron in protein compounds called ferritin. So the ferritin is our storage molecule for iron. So if you do eat a big meal, you eat lots of animal foods or you eat lots of plant foods that contain iron, and then the body has all of this iron coming in, it can stick it on this ferritin and store it in the bone marrow and in the liver and the spleen. On ferritin and then also homocisterin. So it can store it in these compounds so that when the red bone marrow or when we've got some EPO in the system and our body's like, hey, we need to make more red blood cells, then we can go and grab it from the bone marrow where it already is, the liver, the spleen, and go get it off of those ferritin molecules and the homocisterin molecules. Ferroportin transports the iron across the intestinal cell membranes. So when we eat a meal that has heme in it, whether it's heme iron or non-heme iron, when it has iron, the ferroportin transports it. You can kind of remember that because of the portin part. It transports it across the intestinal cell plasma membrane. So it gets it from the middle of our digestive tube across that tube, and then it also gets it from its storage sites into the tissue fluid where it enters the blood. So then when EPO stimulates the production of red blood cells, the iron is released from storage when it's bound to transferrin and carried to the red bone marrow where it attaches to erythrocyte precursors. So the stuff that's going to eventually make the erythrocytes. All right, so that's our number one there is iron as far as our trace elements. And then number two is copper. Copper is also a trace mineral, or a trace element, trace mineral. It's a component of the two plasma proteins. These guys, which we didn't really talk about, but the hephaestin and ceruloplasmin Without these, hemoglobin cannot be adequately produced. So these are some plasma proteins. Remember, there were a whole bunch of them, we said, are in the plasma. These aren't some of the main ones, but they're important to make hemoglobin. And so we need these guys to make hemoglobin, and copper is a part of these two plasma proteins. So if we don't have copper, we don't have these plasma proteins. If we don't have these plasma proteins, we cannot make hemoglobin. If we don't have hemoglobin, we don't have red blood cells. So it's important also that we make sure that we get enough copper in our diet so that we can go through this process to eventually end up with red blood cells. So located in the intestinal villi, Hay-fehs-tin, I cannot say that right, sorry, enables iron to be absorbed by intestinal cells. So that is the important part of this guy here. I'll just say that. Enables the iron to be absorbed, so it's getting it in there. And then the ceruloplasmin, I can say that one easier, transports copper. So it's going to be helping it to move around and helps to create the hemoglobin. So they enable the oxidation of iron to, here we go, the oxidation of iron from Fe2+, sorry, I've kind of written over that, to the Fe3+ here. So that's when it can be bound to its transport protein transferrin. All right, so we have copper, right, which is a component of these guys here. So one located in the intestines, this first one here, enables iron to be absorbed by the intestinal cells. Then this guy here, ceruloplasmin, then transports the copper. And then both of these are able to help the oxidation of iron from the Fe2+ to Fe3+. Then that will be able to be bound to transferrin. And remember, that is how up here, bound to transferrin, and then is carried to the red bone marrow, where it's then able to attach to red blood cell precursors and then be transported where it needs to go. So it has to be able to get to that transferrin. So if we don't have the copper, we don't have these two plasma proteins. If we don't have these two plasma proteins, we don't have absorption of the iron. If we don't have this first plasma protein, we don't have the absorption of the copper. If we don't have this guy, then we don't have transportation of the copper. If we don't have that, either of those, then we don't have the iron being oxidized. If we don't have the iron being oxidized, then it can't be attached to the transferrin. If it's not attached to the transferrin, it can't get to the point where it's attached, where the iron then is attached to the hemoglobin to then make the red blood cells. All right, so hopefully I didn't mess that up enough for you to be able to understand that. So if you look over those notes again, read through the text there, you can kind of follow the process of why copper is important and, of course, why iron is important. So then there's a mention here in the state of copper deficiency. The transport of iron for heme synthesis decreases, and then iron can accumulate in the tissues where it can eventually lead to organ damage. So if you're eating a whole bunch of iron, it's in the body, and then if you then are deficient in copper, you haven't taken in any copper, and then you're not able to move the iron around in order to make hemoglobin, in order to get into the red blood cells, then that can accumulate and can cause some serious issues. The next one is zinc. The trace mineral zinc, just a quick note here, is a coenzyme. Remember, that's something that assists an enzyme in working. It facilitates the synthesis of the heme portion of hemoglobin. So zinc is important to be able to make the heme portion. Remember, the heme portion is that outer portion that then attaches the iron in the center. Then we need some B vitamins. Folate and vitamin B12 are also coenzymes, and they facilitate DNA synthesis. So it's important to have DNA synthesis so that you can make new cells, including the red blood cells. So you need the DNA synthesis in order to make the red blood cells, which means you need those B vitamins for the production of any cells, but also red blood cells. So erythrocytes, red blood cells, live up to 120 days in circulation, after which the worn out cells are removed by a type of myeloid phagocytic cell named a macrophage. So our macrophage or macrophage is going to get it out of there. So it removes it, it takes it, and then it's going to recycle a lot of these things. So these macrophages are located primarily within the bone marrow, the liver, and the spleen. And then the components of these degraded erythrocytes, or the hemoglobin, are further processed. So these macrophages, remember, phage is to eat, and macro is large in size. These are really large cells that eat stuff up. So they go and they seek out damaged cells, worn out cells, including red blood cells, including these erythrocytes, and then they gobble them up and take apart their pieces. And then what they do is they're going to assist in recycling these pieces by breaking them apart. So here is the recycle part. Globin, the protein portion of hemoglobin, remember heme, and then the globin part is broken down into amino acids, because remember it's protein, so protein, when you break it down into pieces, are the amino acids. And then they are sent back to the bone marrow to be used for new red blood cells. The other part of hemoglobin that is not, or the hemoglobin that's not phagocytized, is broken down in circulation. And it releases those alpha and beta chains that are removed from circulation by the kidneys. So some of the macrophages, they'll break them down into pieces, and then the globin portion is broken down into amino acids, and then those amino acids can be sent back to the red bone marrow to be recycled and put into new blood cells. Hemoglobin that's not phagocytized, that's not eaten up and broken down and recycled, is then broken down just in circulation. And then these chains are removed by the kidneys, and that's part of the excretion of the kidneys there. So the iron contained in the heme portion of hemoglobin is stored in the liver or the spleen, primarily in the form of, remember, our ferritin or homocysteine, or carried through the bloodstream by transferrin to the red bone marrow for recycling into new erythrocytes. So we try to use up as much iron as we can. We keep recycling that. The non-iron portion of heme is degraded into the waste product biliverdin. So up here we have the globin portion, right, of hemoglobin. Remember, the heme part and the globin part. The globin part's the protein. We break that down, recycle some of it, get rid of the rest. Then the iron on the heme is recycled. And then we have the non-iron portion of heme, and that is degraded into biliverdin. This is a green pigment, and we'll talk a lot more about biliverdin and bilirubin, which is the next part of the process. It turns it into, which is this yellow pigment, bilirubin. Because bilirubin is then going to bind to albumin and then travels in the blood to the liver. And then the liver is going to change it into and add it to bile. Bilirubin is a part of bile. And we'll talk about that a lot in the digestive system, but quickly that bile is then released into the intestines, and it helps emulsify dietary fats. So when you eat a meal of fats, again, remember our oil and water mixture, the oil kind of sits on top, it's hydrophobic. So we need some way to get that through our system. Part of the way we do that is by releasing bile, and then bile helps to break those fats into tiny smaller pieces that can then be more easily transported in our intestines, and then ultimately will then get into the rest of our system. In the large intestine, the bacteria breaks the bilirubin apart from the bile and converts it to urobilinogen and then into stercobilin. It is then eliminated from the body in the feces. We're going to talk more about those things, the different bilins there. It is important for you to understand, though, that in this section of the text and in this part of the class, that the non-iron portion of heme is then degraded into biliverdin, which is then converted into bilirubin, which is then brought to the liver to put in bile in the gallbladder and stored in the gallbladder. But so that part is important for you to understand. This other part here, in the large intestine portion, we'll talk more about when we get to the digestive system. All right, so then the kidneys will also remove any circulating bilirubin and other metabolic byproducts such as urobilins and secrete them into the urine. So right here, the breakdown of pigments that are formed from the destruction of the hemoglobin can be seen in a variety of situations. And this is just kind of an interesting thing here. So at the site of an injury, the biliverdin, remember from up here, which is kind of a green color, from damaged red blood cells produces some of the colors that we see in bruising. So if you get bumped, you can see some of that biliverdin coming through because of the damage that was done to the red blood cells. The non-iron portion of heme is then degraded into biliverdin because it's in the system, and it gets degraded into this waste product because it's out there floating around. It's been broken off, and then when that happens, it looks green, and it gives us some of the colors that we see with bruising. With a failing liver, then bilirubin, remember that's the other one, after it is biliverdin, it's turned into bilirubin, is not able to be removed effectively from circulation, and it makes the whole body turn into a yellowish kind of color, turns yellowish. And that's seen a lot in some infants sometimes with liver issues. Maybe it's a premature infant that's born, and the newborn isn't able to process things correctly quite yet, and so then they can end up being a little bit yellowish. And that's called jaundice. The stercobilins within the feces then produce the brown color. So up here it talks about the different colors, and that's typically brown in color, and that's us getting rid of it. It's getting, that's our body getting rid of it through feces in this case. And then the yellow of urine is the urobilins. So that's why I'm saying that in this case here, these things are just kind of interesting things to see the different colors and why it is that we see those different colors. So then down here, we can see our erythrocyte life cycle. All right, so let's find number one. Number one's up here. Hemopoiesis of the erythrocytes begins in the hemopoietic bone marrow. All right, so we're talking about the bone marrow here, and it shows you the locations. Remember, we see the iliac crest and parts of the bones here. We have our stem cell, and then we have our erythroblast. Remember, blast is a precursor. Then remember, our immature red blood cell is the reticulocyte. So we have our reticulocyte, which is kind of granular looking because it still has some of the organelles in there. It hasn't purged them all or extruded them all. Then part two here, reticulocytes are released into the bloodstream. You can see that arrow here, into the bloodstream, where they then mature into the erythrocytes, the red blood cells, which then are in the blood for 120 days-ish. Here they are. Then three, the old and damaged erythrocytes are phagocytized. That's this guy here with a macrophage in the bone marrow, liver, and spleen. Then here, number four, the globin, right, the protein portion of the hemoglobin is metabolized into amino acids. You can see that they're kind of showing that it's breaking apart here into pieces, into amino acids, which are reused here. Ba-doop. Hemoglobin protein structure is then broken down into these amino acids. See, there's like the protein portion here, and then they're breaking it into these tiny amino acids, and they are reused. You can see that it's saying this direction. We can go to the heme portion, step 5. The heme portion is broken down into biliverdin. Right, so the heme group here goes to biliverdin, then bilirubin. Right, so biliverdin for transport in the blood. The iron ions bind, so here the iron ions bind to the blood protein transferrin for transport. Go to the liver. Here, remember the liver. Then step 6, the unused heme groups can be recycled and used in hemopoiesis, or they can be converted into bilirubin and used to make bile in the liver. Iron ions can also be transferred to the protein ferritin for storage in the liver. So they're showing the different directions here. It can go over here, back into the red bone marrow, so that we can have some more hemopoiesis. The iron can go into the bone marrow for hemopoiesis. Here, we didn't talk about it a moment ago, but the globin amino acids and the other cell components can also go back into the bone for hemopoiesis. So they can either be stored over here. These guys can either be stored over here in the liver, or they can go over to be recycled and make new red blood cells. So then lastly, in our section about erythrocytes or red blood cells, we're going to talk about some of the disorders of the red blood cells. So the size, the shape, and the number of the red blood cells and the number of hemoglobin molecules are very important. And we've talked about that in different ways, so we're going to just talk more specifically about it. When the number of red blood cells or the hemoglobin is deficient, so there are too few, the general condition is called anemia. We've mentioned that a couple times now. So there are more than 400 types of anemia, and more than 3.5 million Americans suffer from this condition. So we're going to break anemia down into 3 major groups here. So one are those caused by blood loss. So an anemia, remember, so the general condition of anemia is when there is a decreased number of red blood cells or hemoglobin. So if we have a large amount of blood loss, then that would decrease the amount of blood in the body, which would be anemia. One would be then anemic at that point. 2, those caused by faulty or decreased red blood cell production. So if we have a decrease in red blood cell production, we're not making enough red blood cells, or if the red blood cells that we're making are faulty, then that's another type of anemia. And those caused by excessive destruction of red blood cells. So if we are making the red blood cells and we haven't lost blood in some form, if there is an increased breakdown of red blood cells for some reason, then that would be anemia also, because we still wouldn't have enough of red blood cells. Even though they're being produced, they're being broken down, so we're not able to use them. So clinicians often use 2 groupings in diagnosis. They use the kinetic approach, which is evaluating the production, destruction, and removal of the red blood cells. And then the morphological approach looks at the red blood cells themselves. So looking at their size, looking at if they're being formed correctly, looking at making sure they have all their parts. So a common test is the mean corpuscle volume, or MCV, which measures size. So normal size cells are normocytic. Smaller than normal cells are microcytic, and larger than normal cells are macrocytic. That should be pretty easy to remember. Normal, cytic, and then micro meaning smaller cytic, and macro meaning larger cytic. Reticulocyte counts are also important. Remember, we talked about that as being a stage in the process that can give us an indication of how red blood cell production is going. The effects of anemias are widespread because reduced numbers of red blood cells or hemoglobin will result in lower levels of oxygen being delivered to body tissue. So if you can imagine, if there's just an overall decrease in red blood cell or decrease in hemoglobin, that means a decrease in oxygen to every single cell in the body. So we can see a lot of different issues, a lot of different effects, a lot of different symptoms. So some of the symptoms that are kind of generalized that's seen, because if we can imagine, if we have a decrease in oxygen, we'll have some fatigue, lethargy, an increased risk for infection. An oxygen deficient brain impairs the ability to think, may prompt headaches, irritability, can leave patients short of breath if we don't have enough oxygen there, even as the heart and lungs are trying to pump more and more and more blood. So the first group, the blood loss anemias, as it says here, are fairly straightforward. So in addition to bleeding from the wounds or other lesions, these forms of anemia can be due to ulcers. So if there's bleeding in the stomach, that's an ulcer. There's tissue that's bleeding in there that's losing a lot of blood. Hemorrhoids that can bleed when there is a bowel movement specifically. Inflammation of the stomach, so if things are inflamed, there can be a lot of blood loss there, a lot of scraping and tearing of the tissue and loss there. And some cancers of the GI tract or gastrointestinal tract, that's the digestive tract. So there would be a lot of blood loss there, probably mostly in the feces. Excessive use of aspirin or other NSAIDs, non-steroidal anti-inflammatory drugs like ibuprofen, can trigger ulceration, so they can give ulcers, it says so on the packages even, and gastritis as well. So that can cause a problem and an increase of blood loss. Excessive menstruation, we've talked a couple of times now about how females tend to have a little bit lower blood cell count, a little bit lower hemoglobin or oxygen count, and part of that is due to menstruation. So if there's excessive menstruation, that's going to be a lot of blood loss, resulting in anemia. Also, loss of blood during childbirth. So that's another example. So anemias that are caused by faulty or decreased red blood cell production. We talk about sickle cell anemia. That's a very common one you perhaps have heard of. Also iron deficiency anemia, vitamin deficiency anemia, and diseases of the bone marrow and the stem cells. So all of these things leading up to a healthy red blood cell, because then that would cause our faulty or decreased red blood cell. So a change, you can see here, we're going to talk about sickle cell disease here for a moment. So sickle cell disease or sickle cell anemia is this disorder where a red blood cell looks sickle shaped, kind of moon shape, a sickle shape. There you can see one I pointed to at the moment. It's a genetic disorder. It's caused by the production of an abnormal type of hemoglobin. So it's called hemoglobin S is made, and then that delivers less oxygen to tissue. So it looks this way. It looks like this sickle shape, and that's due to the hemoglobin. It affects the shape of the red blood cell, and then it is unable to get as much oxygen to the tissues. It causes this shape. So these abnormally shaped cells can also then become lodged in small capillaries. Remember we talked about these red blood cells needing to get to these really tiny areas, and they'll fold and bend on themselves to get in there and then get the oxygen to those tissues, and then they'll make their way out as well. But in this case they can actually become lodged in there. Because they're not able to fold on themselves and squeeze through. And then that can end up blocking blood flow to tissues and then cause some serious problems, some painful joints, delayed growth, in this case seriousness of blindness or cerebrovascular incidents, strokes. So sickle cell anemia, again, is a genetic condition, but it's often found in individuals of African descent. So we do see that in that area. So people will look at that when physicians see these kinds of things like the fatigue, the lethargy, the headaches, the irritability, all of these things, and they have a patient of African descent, they may take a look at that as one of the reasons. So then down here we have our iron deficiency anemia. And this is the most common type and results when the amount of iron is low. So this is obviously what it says. So iron is deficient. You're not getting enough iron in the body. Then if you're not getting enough iron, again, you're not able to produce that heme. Therefore, you're not able to produce hemoglobin and not able to produce the red blood cells. So it can occur in individuals with a deficiency of iron in the diet. It's common in teens, children, vegans, and vegetarians. So teens and children, mostly because perhaps they're not eating a very well-rounded diet. Teens perhaps aren't eating a very well-rounded diet, and then in children, sometimes it may be difficult to get children to eat certain things, so then it might not be very well-rounded. In vegans and vegetarians, because again, remember, the non-heme iron is not absorbed as well. So it's important for somebody that's a vegan or a vegetarian to pay attention to the foods that they're eating and make sure they get a well-rounded diet in order to make sure that they get enough iron. So additionally, the iron deficiency anemia can be caused either by an inability to absorb or transport iron, or slow chronic bleeding. So in this case, it would be a slow bleeding, not like that first group that was talking about like a huge gush of blood, a huge injury that would just flush all of the blood out. But in this case, just a slow chronic bleeding that then would not allow the body to recycle the iron. So our body counts on that iron to be recycled, as well as an intake of iron from the diet. Another one here, the vitamin deficient anemias, generally involve insufficient vitamin B12 and folate. So remember, those were important to producing the red blood cells. So when we're talking about a deficiency in vitamins, then those are going to be two important ones. Again, not getting it in the diet. And the vitamin B12 a lot of times is seen in vegans and vegetarians, again, if they're not paying attention to making sure they get enough vitamin B12. That said, even though people have heard a lot about vegans and vegetarians not getting enough B12, and that might be on a person's mind if they are vegan or vegetarian, a lot of people that do eat animal products also are being found to be vitamin B12 deficient. A lot of times people think it's because the bacteria that produce or have the vitamin B12 in them is not as prevalent nowadays as it used to be when we have more of these farming practices being changed. So it's important to look at that if a person is having some of these issues related to anemia. Megaloblastic anemia, another one, involves a deficiency of the vitamin B12 and or folate. Involves diets deficient in these, again, lack of meat or a viable alternative source. One thing I didn't mention just a moment ago when I mentioned vegans and vegetarians is this overcooking or eating insufficient amounts of vegetables may lead to a lack of folate. So even if a person eats lots and lots of vegetables, if they are cooking them or boiling them to mush, then they may still not have these things because they're destroying the vitamins before they're able to even eat the food. So they may not have it. Pernicious anemia, right here, the next one is caused by poor absorption of vitamin B12, often seen in patients with Crohn's disease. This is an intestinal disorder that causes poor absorption of vitamins. One of those vitamins is B12. So in this case, surgical removal of the intestines or stomach happens sometimes, and then can cause poor absorption of B12. So not only with Crohn's disease, but also sometimes people get parts of their intestines or stomach removed, sometimes seen in weight loss surgeries. Those people, as well as people that have intestinal parasites or AIDS, will really have to pay attention to their B12 intake so they don't get pernicious anemia, because that's important, again, in the production of the red blood cells. Pregnancies, some medications, excessive alcohol, and some diseases such as celiac disease are also associated with vitamin deficiency. So a lot of these last ones here are talking about the deficiency of vitamins, and so it's kind of seen as one big group, vitamin B12 and or folate deficiency, and then there are various reasons for that. So diet's deficient, you need to make sure you're getting that in your diet. Pregnancy, you need to make sure you're getting enough when you're pregnant. If you have medications that tend to leach B12 or folate from the body, then you need to make sure you're compensating in diet. Excessive alcohol consumption, alcohol really messes up the vitamin balance, so you need to make sure you're getting enough in the diet. Celiac disease can really cause some issues with people. They stay away from certain foods, and then that can lead to a decrease in some of the vitamins. So all of these things are mostly related to the body kind of needing more due to these different reasons, and so it's necessary for one to make sure that they're taking enough in their diet. Here, assorted disease processes can also interfere with the production and formation of red blood cells and hemoglobin. So if myeloid stem cells are defective or replaced by cancer cells, there will be insufficient quantities of red blood cells produced. Remember, the myeloid cell is one of those stages that you have to get through. A myeloid stem cell is then going to eventually lead to that erythrocyte. So if there are some diseases that cause problems with that myeloid cell, whether it's defective or replaced by a cancer cell, then you won't get the red blood cell production that's needed. Aplastic anemia is the condition where there are deficient numbers of red blood cell stem cells. So aplastic anemia is often inherited. So since it's at the stem cell level, not necessarily like you didn't get enough iron level, a lot of times that's inherited, or, as mentioned here, triggered by radiation, a medication, chemotherapy, or infection. So those are things that need to be looked out for. Thalassemia is an inherited condition, typically occurring in individuals from the Middle East, the Mediterranean, African, and Southeast Asia. And this is where the maturation of the red blood cells doesn't proceed normally. Remember, it goes from a reticulocyte to an erythrocyte. And so there's a problem there, and when it's very, very severe, it's called Cooley's anemia. Lead exposure from industrial sources or even dust from paint chips of iron-containing paints or pottery can lead to the destruction of the red bone marrow, which would then lead to a decrease in red blood cell production. Various disease processes also can lead to anemias. Chronic kidney disease can lead to a decrease in EPO production, right? So if you have problems with your kidney, remember your kidney is where you release erythropoietin, which then causes the production of red blood cells. If there's a problem with the kidney, you don't have EPO, you don't have red blood cell production. hypothyroidism, some forms of cancer, lupus, and rheumatoid arthritis. So the opposite of that, we've been talking about anemias, which are the decrease in red blood cell production. The opposite, which we've mentioned several times now, polycythemia. So this is an elevated red blood cell count. So when there is too much red blood cell in the body, too many red blood cells, is detected in a patient's elevated hematocrit. So we saw that earlier. We saw the picture of the hematocrits, and we saw that there's a lot more red blood cell area in that hematocrit when there was polycythemia. It can occur transiently in a person who is dehydrated. So transiently meaning it's not an ongoing condition. It's just something that happens sometimes. For example, when somebody hasn't had water for several days for whatever reason, they're very dehydrated. That would mean they have lower blood plasma, which would then look like they have more red blood cells, because we're just talking about the amount of red blood cell compared to the whole. So if there's less blood plasma because there's less fluid, less water in the body because they're dehydrated, then there's more hematocrit, a larger hematocrit. That's transient polycythemia. So for reasons mentioned earlier, a mild form of polycythemia is chronic but normal in people living at high altitudes. We talked about that with the altitude sickness, but people that live there have that, a mild form. Some elite athletes train at high elevations specifically for this. Remember, rather than doping or increasing the EPO in their body artificially, they'll maybe go to a higher elevation so that their body naturally does it, and then they come down to the lower elevation to do the sport. Finally, a type of bone marrow disease called polycythemia vera causes an excessive production of immature erythrocytes. So polycythemia vera can dangerously elevate the viscosity of the blood. Remember, when we have more red blood cells, that's thickening the blood, which can then raise the blood pressure, and then it makes it really difficult for the heart to pump the blood throughout the body. So again, when it's transient, this may be an issue for a day or so, and then the blood can thin out a little bit more and the heart can relax and it won't be so difficult. But if this is an all the time kind of thing, as in polycythemia vera, then this can be a severe problem. It is relatively rare and occurs more often in men than in women, and is more likely to be present in elderly patients over 60 years of age. So that's a with age kind of thing, where the body is less able to keep balance, maintain homeostasis. All right, so that is all about the erythrocytes. And then we're going to next move on to the leukocytes and platelets.